Hello children, hope you are doing great, and we are in the last week of our preparation for the coming NEET, and every minute matters for you, and we know that pretty well. So what we are going to do now is we are going to revise the entire syllabus of both first and second year into 4 units, and today is the first day of that 4 chapters, 4 units. We are going to get rid of living world, biological classification, and plant kingdom. So your job is to listen to me for 45 minutes, and I'm going to discuss the most important lines which are there in the textbook with regard to these 3 chapters. So listen carefully and see that you don't make any errors in the coming exam without wasting a single minute. We'll start with the first chapter called Living World. Number of questions asked in Living World for the past many years, for the past 5 years, 1. Sometimes in 1 or 2 years there was no question asked from Living World. But again, all right, this is an important chapter. One question is a definite question from Living World. We are going to spend 10 minutes with this chapter called Living World. Listen carefully. All the living organisms share similarities both horizontally and vertically. They resemble their peer group and also to their ancestors. Anthropocentric, human-centric. And the next important, what is the meaning of living? Technical meaning of living is those which has got metabolism, growth, reproduction, consciousness, and cellular nature. That is the meaning of living. Those which doesn't have all this is said to be non-living. Now, first, growth. We'll spend 2 minutes here. Growth in non-living is extrinsic. Growth in living is intrinsic. Growth in plants, indeterminate. Growth in animals, determinate. Dead organisms do not grow. Growth is not a defining feature. The meaning of defining is it should be unexceptional. Growth is not a defining feature because it is seen even in non-living, extrinsic, increase in mass. Growth in living is determinate, and growth in living is intrinsic, and it occurs both in increase in mass and number. Chalo. Next is reproduction. Not many points are in fact new to you. Reproduction, alga, fungi, and protoneomorphosis. They reproduce asexually by fragmentation. Bacteria divide by binary fission. Amoeba divide by binary fission. Diatoms, dinoflagellates, they also divide by binary fission. Then you have got budding. Yeast is going to reproduce by budding. Planaria is going to reproduce by true regeneration. So every separated part is capable of developing into entire organism. Again, reproduction is not a defining feature because there are some living organisms which don't reproduce, mules, worker bees, etc. And more importantly, in Monera and Protista, reproduction and growth are synonymous. In 2 kingdoms, it is the same. That's it. Next is metabolism. The sum total of anabolic and catabolic changes occurring in an organism is said to be metabolism. For metabolism to occur, cells are needed. So therefore, metabolism is seen only in the 5 kingdoms of Whitaker's classification. Viruses do not have cells, therefore there is no metabolism. Metabolic reactions could be anabolic, constructive, photosynthesis, or catabolic, respiration. Next, metabolic reactions occurring inside the body of an organism is said to be metabolism. Metabolic reactions occurring outside the cell in a cell-free system are said to be living reactions. Wonderful example for living reaction is the cell-free system of protein synthesis done by Marshall Nirenberg in deciphering genetic code can be a wonderful example for living reaction because this reaction is occurring outside the cell, not inside the cell. Then we talk about consciousness. The meaning of consciousness is response to stimulus. All living organisms starting from Monera to Animalia respond to stimulus. Mind you, sense organs are not obligatory for that. Your textbook says it is the most obvious and technically complicated because all living organisms do not respond similarly to the same stimulus. Consciousness is seen in all organisms. Photoperiod means the light period have an effect on reproduction both in plants and animals. You call it as photoperiodism. So we all respond to stimulus. That is an important point. The next one, self-consciousness. Ability to know about themselves, to be aware of themselves is called self-consciousness. Only humans are going to possess self-consciousness. They have neurocontrol system, therefore they have self-conscious, they are aware of themselves. All right, I normally give one example here. All right, a dog and a dog trainer. Imagine if there is an explosive there. Dog and dog trainer both are conscious that there is something which is fishy there. Both of them are conscious. But the dog trainer holds himself back and sends the dog because he knows pretty well the danger of that explosive. So it means that self-consciousness is seen only in humans, and whereas consciousness is observed in all. Brain dead people do not have self-consciousness. Last important point. The property of a tissue is because of interaction of cell. Property of a cell organelle is because of the interaction of the chemical constituent present in that cell organelle. So all these emergent properties is because of these interactions. That completes the first important part. Then we are in the last 5 minutes. I told you we are not going to take much time here. The last 5 minutes is about biodiversity, the concept of taxonomy. Then you heard in your zoology part that Robert May, he reported almost 7 million species as a biodiversified number. Out of 7 million species, we were able to identify only 1.7 to 1.8 millions. It means that much of the species are yet to be identified. What is the technique? So first you have to characterize, then you have to identify, then you have to name, and then you have to classify. All this put together is said to be taxonomy. Identification, for that we require taxonomic aids like herbarium, flora, botanical gardens, museums, etc. Then we have got nomenclature, naming of the plants. Linnaeus was the first scientist who reported that naming of plants should be in 2 words, genus and species. And later, ICBN rules have endorsed Linnaeus' concept of 2 words and set some other rules. What are the rules of ICBN? Very popular area. 1. Naming of plants should be in 2 words, genus and species. 2. The scientific name should be published in italics. All right, because it is derived from Latin. 3. If it is not published, if it is handwritten, the genus and species is separately underlined, not the scientist. Be careful. And even the scientist is not printed in italics. That's also an important point. The scientist's name written after the species should neither be printed in italics nor should be underlined. It should be an abbreviated form, a short form, LINN for Linnaeus. Genus name should start with a capital letter and should be a noun form. And the species name should start with a small letter and should be an adjective form. These are the these are the points about nomenclature. Then we talk about classification. There are 7 taxonomic categories starting from species to kingdom. That is said to be taxonomic hierarchy. When you move from species to kingdom, the number of common characters decreases. That is going to make the classification part more complex. What is the meaning of taxonomic category? That rank or division which helps us to classify an organism is taxonomic category. Taxon is an existential unit of the taxonomic category. Say for example, if you say genus, genus is a category. The existential form Solanum is said to be taxon. Well, you know pretty well about that. So we are going to spend 2 minutes for all, starting from species. Before I talk about that, systematics. This term was coined by Linnaeus. The term, it is derived from Latin. Systematics is the classification of an organisms based on phylogeny. Systema Naturae is also a book written by Linnaeus where he named all the animals. Just like Species Plantarum, he named all the plants. Now I come back to taxonomic categories. Species. Ernest Mayr, who is called Darwin of 20th century, made species concept, gave the definition of species. All the organisms which are morphologically similar and which can freely interbreed is said to be species. Species is the smallest unit of classification. It is also an existential unit of classification. Species name should always start with a small letter. Very important, very simple, you know pretty well about that. Then we go to genus. A group of species put together is kept under genus. Genus name should start with a capital letter. Genus, say for example mango, it is Mangifera. For wheat, the genus is Triticum. A genus can have 1 species epithet, or a genus can have more than 1 species epithet. Solanum, tuberosum, Solanum, melongena, Solanum, lycopersicon, Panthera, Leo, Panthera, pardus, Panthera, nigris. So these are tigris. These are all the examples of different what is called the species. A genus can have more than 1 species. Well, this is very simple. The next one what we have is family. Plant families ends with AC and animal families ends with day, a day. Say for example, mango belongs to family Anacardiaceae. Wheat belongs to family Poaceae. housefly, all right, then housefly, and we have got humans, hominidae, all right, Mucidae, housefly. So all of them ends with day. That's a very easy way to remember. All right, a group of genera with a common characters are grouped under family. Convolvulaceae, Solanaceae, they're also families. Then after families, then what we have is class. Sorry, order. All right. Now from here onwards, the aggregate of characters is taken as a criteria. Now imagine mango belongs to order Sapindales, wheat belongs to order Poales. Easy way to remember. All right. So then don't forget, Primata is the order of humans. Diptera is an order of housefly. Order ends with ales as far as plants are concerned. Sapindales, Polymoniales, this point is important. Solanaceae and Convolvulaceae are put under what is called order Polymoniales based on floral characters, is a very popular question. Then after order, then we have got class. Then wheat belongs to class monocots, mango belongs to class dicots. That's about the class children, which is easy way to remember. Insecta is the class of what is called housefly. All right, then we have got what is called division. If it is plants, phylum if it is animals. All right, you know that division angiosperms, mango and wheat, both of them belongs to division angiosperms. And the housefly and all that, with regard to animal kingdom, you're pretty sure about the division there. Then we talk phylum, we call arthropoda. All right, then we have got next is kingdom, plant kingdom and animal kingdom. Just now I told you, when you move from what is called species to kingdom, the common characters decreases and the complexity of classification increases. Taxonomic categories are not mere morphological entities, not mere morphological aggregates, but distinct biological entities is a point you have to remember. That completes the living world, children. One question is normally asked, don't need to read taxonomic aids. Herbarium is a word, is an aid used for plants. Flora is also used for plants. All right, botanical garden is also used for plants and useful for identification of plants. Museums for both plants and animals. Zoological parks, all right, is only for animals, and manuals for identification for both plants and animals. Anyway, these are not questions to be asked. That completes the living world. This is the max I think your examiner can ask you. I expect question 1, it could be either from nomenclature, rules of nomenclature, ICBN rules, or anything from taxonomic categories. That is the most popular area from where the questions can come. Chalo. Now the next 20 minutes we are going to allot for biological classification, just 20 minutes. We have got two types of classification, non-scientific and scientific. Based on utility, if you classify organisms, primitive man, he was classifying useful plants and carrying them along with it, and not useful he ignored. So that is a primitive non-scientific classification. Scientific classifications we have got 3, Aristotle, Linnaeus and Whittaker. Aristotle is the first scientific classification where he took morphology of stem as a criteria for classifying plants into herbs, shrubs and trees. He even classified animals, those with red blood and those without red blood. Next is Linnaeus. He classified all living organisms based mainly on the presence and absence of cell wall. Those with cell wall he called them as plants, those without cell wall he called them as animals. Bacteria he called them as plants because they contain cell wall. Diatoms, dinoflagellates, he called them as plants because they contain cell wall. Fungi he called them as plants because they contain cell wall. All right, and protozoans he called them as animals because they don't have cell wall. All right, so this is how the Linnaeus classified. The disadvantage of Linnaeus classification is both autotrophs and heterotrophs are put together or kept together. Unicellular and multicellular are kept together. All right, prokaryotes and eukaryotes are kept together. And this classification is based on what is called some superficial characters. Therefore, this does not represent a correct classification as such. Then comes Whittaker, who actually smartly worked on the demerits of Linnaeus and classified 5 kingdom classification. Prokaryotes and eukaryotes are not divided, that is Linnaeus. Then what Whittaker did, he divided prokaryotes and kept under kingdom Monera. Unicellular and multicellular are not separated is a demerit of Linnaeus. Then he separated unicellular eukaryotes and kept under kingdom Protista. Fungi are kept under plants, that was the biggest demerit of Linnaeus. and fungi separated thoroughly and gave a separate kingdom by Whittaker. Cell structure, body organization, nutrition, reproduction and phylogeny are the 5 parameters on which Whittaker classified the 5 kingdoms. Now, what are the questions which can cast from the table? Unicellular, Monera protista, tissue grade, fungi, plantia, animalia. Reproduction and growth are synonymous, Monera protista. They are not synonymous, fungi, plantia, animalia. Prokaryotic, only Monera. Remaining 4 kingdoms, eukaryotic. Organs, plants and animals. Organ systems, only animals. Nutrition, if you look, Monera, autotrophs and heterotrophs. There is a huge nutritional diversity in Monera. Photoautotrophs, chemoautotrophs, photoheterotrophs, chemoheterotrophs. Coming to protista, autotrophs and heterotrophs. Diatoms, dinoflagellates, euglenoids are autotrophs. Slime molds and protozoans are heterotrophs. Then coming to fungi, heterotrophs, exclusively heterotrophs. Coming to plants, exclusively autotrophs. And coming to animals, exclusively heterotrophs. One point you have to remember, saprophytic nutrition is seen in almost all 4 kingdoms except plantia. Holozoic, ingestive mode of nutrition is seen not only in animals but some of the protists like euglena. Then chemosynthetic nutrition, chemoautotrophs, that is seen exclusively in Monera. Remember that. So these are all what is called chemoautotrophic, I mean to say. Chemoautotrophic nutrition is seen exclusively in Monera. So that completes the nutrition part, so don't make any errors here. Then coming to the next important one, what we have is about reproduction and phylogeny. Monera is kept first and Animalia is kept last because Animalia evolutionarily more advanced when compared to the remaining kingdoms. It's okay. Now we start without wasting any time. What are the organisms which are kept in which group by Linnaeus and Whittaker? Let us look. Bacteria. Linnaeus classification, they are kept under plants because they contain cell wall. In Whittaker, Monera. Okay. Then diatoms and dinoflagellates. In Linnaeus classification, plants because they contain cell wall. Whereas in what is called Whittaker's classification, they are kept under protista. Chlorella and chlamydomonas. Watch out for this question. Chlorella and chlamydomonas are kept under plants by Linnaeus because they contain cell wall. Because they are unicellular eukaryotes, they are kept under protista by Whittaker. Very important. You have to remember this. Then we have got slime molds, euglenoids. Linnaeus did not classify them. Alright, whereas Whittaker kept them under protista because they are unicellular and eukaryotic. Protozoans, Linnaeus put them under animals because they don't have a cell wall, whereas Whittaker kept under protista because they are unicellular eukaryotes. So this is the important, and of course fungi, they are kept under plants by Linnaeus because they contain chitin, a cell wall material, and they are given a separate kingdom called fungi by Whittaker. So the 2 kingdoms which are common in almost all classifications is plants and animals, even though the organisms kept in these keep on differing from one classification to another. So the introduction part is very important, children. I covered almost all points. Alright, now it is for you to remember all this. Now coming to Monera. How much time we are going to spend here? 5 minutes, not more than that. Look here, how to remember this. Sorry. Monera. Archaebacteria and eubacteria, first division. Next, archaebacteria is again divided into methanogens, thermoacidophiles and halophiles. And I want to tell you only 2 points. Archaebacteria contain pseudomurine in their cell walls. It can keep this archaebacteria to survive in hostile environments, asked many times in NEET. Archaebacteria used in biogas production is methanogens, thermoacidophiles, hot springs, halophiles, they grow in salty areas. Alright, archaebacteria have caused some very important characters like presence of histones, 3 types of RNA polymerases, that's what you studied, which is the character of eukaryotes. So it has got some eukaryotic characters. That is the reason Carl Woese have separated archaebacteria and gave a separate kingdom called archaebacteria, and remaining bacteria he kept under eubacteria. So archaebacteria are very distinct to some extent, alright, and they have shown some differences with respect to eubacteria as well. Chalo, archaebacteria is over. Now we go to eubacteria. Eubacteria is divided into 2, autotrophs and heterotrophs. Alright, first I want to get rid of heterotrophs. Heterotrophs can be saprophytes, example lactobacillus, can be parasites, example xanthomonas citri, can be symbionts, example rhizobium. Put that aside now. Now I come back to autotrophs. Autotrophs are again divided into 2 types, photoautotrophs and chemoautotrophs. Example for photoautotrophs is cyanobacteria. Example for chemoautotrophs is iron bacteria, nitrifying bacteria, sulfur bacteria, etc. What is the difference between photoautotroph and chemoautotroph? Both of them reduce atmospheric carbon dioxide to glucose. That is the reason they are called autotrophs. If it occurring in the presence of light, photoautotroph, and this carbon dioxide to glucose require energy, and if that energy is because of the oxidation of inorganic chemicals, then you call it as chemoautotroph. Iron bacteria, they oxidize iron, get energy, and that energy is responsible for converting CO2 to glucose. And nitrifying bacteria, iron bacteria, sulfur bacteria, they're all examples for chemoautotrophs. That does not show photosynthesis. Example for photoautotrophs, cyanobacteria, green sulfur bacteria called chlorobium, purple green sulfur bacteria called chromatium. What did Von Neil said? In green sulfur and purple sulfur bacteria, it is the H2S which get oxidized, not water, and oxygenic photosynthesis, you have to remember. Now I come back to the last cyanobacteria. I'll tell you 5 points. They are unicellular, filamentous, or colonial. 2, they are both terrestrial and aquatic, primarily aquatic. 3, they are surrounded, the filament is surrounded by a mucilaginous sheath. 4, they contain heterocysts, which are nitrogen fixing cells, nostoc, anabina, oscillatoria. That is the reason they can also be used as biofertilizer. 5, they form water blooms, they stay on the surface of water. 6, they show oxygenic photosynthesis, H2O gets oxidized, oxygen is liberated. 7, they can be used as biofertilizer. 8, they doesn't show any sexual reproduction as such, they show only asexual reproduction. So these are the important points about cyanobacteria. Also remember, spirulina is a cyanobacteria which is used as SCP. Don't get confused between spirulina and spirogyra. Spirogyra is a green alga which is multicellular with isogamy and non-motile gametes. This is spirulina used as SCP. That's it. That completes Monera. I forgot 3 points, children. They're simple in structure, complex in behavior. Next important point, they have nutritional diversity. Third important point, they show primitive type of sexual reproduction where there is a transfer of DNA from one bacterium to another, either with the help of a conjugation canal produced by sex pili, you call it as conjugation, or with the help of an external medium, you call it as transformation, or with the help of a virus, you call them as transduction. Either way, it is the transfer of DNA from one bacterium to another which brings variations in a bacterium which receives it. Therefore, it is a sort of sexual reproduction. Bacteria can also reproduce asexually by endospores. You heard about sporolac? Alright, whenever you had a dehydration issue, your mother used to add a powder to water, and that is sporolac. What is sporolac? It is lactobacillus. It is probiotic. This sporolac, when it enters into your body, this lactobacillus is going to fight against disease causing microbes and eliminate them. It is a type of biocontrol agent. It's a type of biological control which is observed in your stomach. That is endospore. Sporolac in that packet contain lactobacillus which stay in the form of endospore. Anyway, that completes Monera. We are trailing by 10 minutes, so we have to find out a way to move faster. Next we go to Protista. Points about Protista, they are mostly aquatic. Yes, the boundaries are not properly fixed. Yes, first time you see zygote formation. Yes, they are unicellular. Yes, they are unicellular and eukaryotic. Yes. What are plant protists? Diatoms and dinoflagellates, because they resemble plants a lot. What are plant animal protists? Euglenoids, because they show characters of both. What are plant animal fungal protists? Slime mouse. And what are animal protists? Protozoans. Okay. Now we'll start with plant protists, diatoms and dinoflagellates. We'll talk both of them together. First, habitat. Dinoflagellates, red tide, all of you heard about red tide, goniolax. Red tide is not seen beside your house in a river. No, it is seen only in sea. So it's very clear that dinoflagellates are marine. So diatoms, they are both marine and freshwater. Remember that. Next, cell wall. Cell wall of diatoms contains cellulose along with silica. The silica is indestructible and form kieselguhr once it settles at the bottom. After the diatoms die, it is used in polishing, filtration, etc. Cell wall, don't forget silica. Alright, along with cellulose. Cell wall of dinoflagellates have got thick armor of cellulosic plates. Next one, flagella. Diatoms do not have any flagella, they show gliding movements. Whereas dinoflagellates contain two flagella which are perpendicular to each other. Next, diatoms have chlorophyll A and C, and other pigments are very less. Whereas dinoflagellates contain chlorophyll A and C with huge amount of xanthophylls, therefore they are variously colored. Asexual reproduction in both is binary fission. And the most important point about diatoms is diatomaceous earth, you remember that? They may be pinnales and centrales, all that is not that much important. And for dinoflagellates, remember goniolacs, which cause red tides. Coming to euglenoids. They are plant animal protists. They contain pellicle, 2 flagella, 1 short and 1 long, which show 9+2 arrangement, which you don't see that in Monera. In Monera, there are no 9+2 arrangement of microtubules. It is the flagellin proteins which is arranged in flagella. You know pretty well about that. First time in Protista, you see 9+2 arrangement of microtubules in the flagella. Pellicle is present. Most importantly, it contains chlorophyll A and B, just like higher plants. In the presence of light, it shows oxygenic photosynthesis. In the absence of light, it predates on small worms. It shows holozoic nutrition. Holozoic nutrition is not only seen in animals and some members of Protista. Example is Euglena. Because of 2 types of nutrition, we say that it shows mixotrophic nutrition. Palmella stage and all that is not that much important. This is a very important point. So that is plant animal. It doesn't have any cell wall animal character. It have got chlorophyll A and B plant character. Therefore, we rightly call plant animal protists. Then we talk about the next group called slime molds. We are not going to take much time in slime molds. This is the amoeba of plus strain. This is the amoeba of minus strain. I want to say mixed amoeba. Mixed amoeba. And these 2 amoeba fuse to form a diploid cell. You can call it a zygote. This diploid cell is going to increase in its size, and this nucleus which is diploid undergo many free nuclear divisions. This vegetative mass, which is unicellular, multinucleate, and which grow luxuriantly whenever there is availability of dead organic matter, it's a saprophyte, that is said to be plasmodium. It is unicellular and multinucleate. Now what happens to this plasmodium? Under favorable conditions, it spreads on the substratum. When conditions are not favorable, what happens to this plasmodium? This plasmodium is going to produce what is called a fruiting body called sporangium. The fruiting body is called sporangium. All these diploid nuclei migrate into sporangium. They undergo reduction division and give rise to spores. Interestingly, the spores are covered by a wall. The spores are covered by wall, and the spores get liberated from here. Once the spores get liberated from here, spores covered by wall, plant character. Once the spores get liberated from here, 50% of the spores will develop into plus strain mixed amoeba. The 50% of the spores develop into minus strain mixed amoeba. Slime molds are going to show algal, fungal, and slime molds are going to show plant, animal, and fungal character. What is the plant character? Presence of cell wall around the spores. What is the animal character? Nowhere you see a cell wall anywhere except in the spores. Alright, then what is the fungal character? Presence of sporangium, production of sporangium, fruiting body, spores, they are all typical fungal characters. That's about slime mold, children. I explained nicely. Next one, what we have is protozoans. We have got amoeboid, example is entamoeba, flagellated, trypanosoma, which cause sleeping sickness, ciliated, don't forget paramecium. It is binucleate, don't forget that. It has got plenty of cilia. It can show active movement. That point is equally important. Then you have got sporozoan. A spore-like stage is seen in a malarial parasite called Plasmodium. All of them are protozoans. In lineage classification, mind you, they are kept under kingdom Animalia because they don't have a cell wall, whereas in Whittaker's classification, because they are unicellular and eukaryote, therefore they are kept under kingdom Protista. Chalo. Now we have got the next important unit called fungi. And in fungi, children, without wasting any time, this we are going to start with Phycomycetes. I want to tell you 5 points about Phycomycetes. 1, they are mostly aquatic. 2, they are unicellular and multinucleate. Remember, if you take for example Rhizopus, it is unicellular and multinucleate. It is unicellular and multinucleate. Don't forget this. 3, asexual reproduction occur with the help of spores which are produced inside this bag. It could be non-motile spores called sporangiospores, or it could be motile spores called zoospores. Both of them are produced inside. They are endospores. So asexual spores are endospores, non-motile aplanospores or sporangiospores, motile zoospores. Next, sexual reproduction. These hyphae can produce gametes. Gametes fuse, plasmogamy and karyogamy occur fast. Alright, both of them occur together to form a diploid zygospore. Your textbook says oospore is the other name for zygospore, correct? Alright, and this zygote undergo reduction division and give rise to germ spores, which are sexual spores. Rhizopus, bread mold, Don't forget that. Albigo, very popular question, which cause white rust in mustard. These are the 2 points. That is about what is called a group called Phycomycetes, and the most important cynocytic, unicellular, zoospores, isogamy and oogamy. The diploid cell is zygospore. Next one what we have is Ascomycetes. We'll spend 2 minutes here, not more than that. These are the ascospores of plus strain, sexual spores. These are the ascospores of minus strain. Ascospore of plus strain develop into a mycelium, and this mycelium is said to be a primary mycelium. The terminal cell is said to be antheridium. This is also a mycelium produced from this spore. This is the primary mycelium, which is monokaryotic. The terminal cell is said to be ascagonium. These are the sex organs. So these are the ascospores. This is the primary mycelium of plus strain, primary mycelium of minus strain, antheridium and ascagonium. What happens here? Plasmogamy occur. Protoplasm fuse, but not the nuclei. This nuclei migrate, but that doesn't fuse. So plasmogamy occur, but not karyogamy. What happens to this diploid? I'm sorry, it's not diploid. What happens to this dikaryotic? It is haploid only. What happens to this dikaryotic cell? It develops into a mycelium. It is going to develop into a mycelium like this. This mycelium, what you're going to see here, is naturally dikaryotic. It is cup-shaped, as you could see. It is cup-shaped mycelium. This is called mushroom. Children, there is no other mushroom. This itself is a mushroom. These are costly mushrooms, morels and truffles. Alright, this is also called ascocarp. This is the cup-shaped mushroom which is edible. Mushrooms are rich in proteins. It is also an SCP. Alright, so therefore, this is the mushroom, ascocarp. The terminal cell of this is said to be ascus, which is sac-like. Hence, it is also called sac fungi. Now the 2 nuclei fuse. Look where plasmogamy is occurring here. Look where karyogamy is occurring here. Do you see a long time gap between plasmogamy and karyogamy? Yes, that is the result. That results in the formation of a dikaryotic mycelium. Very popular question. What happens next? It undergo reduction division and produce ascospores. Are they produced inside or outside? Inside. Are they sexual spores? Yes. And these 4 spores get liberated, and this some of them plus and some of them minus. The remaining story is known to you. Ascomycetes can also reproduce asexually by conidiospores. The difference, ascospores are endogenous, conidiospores are exogenous. They are present on the hyphae. And mind you, conidiospores are not only seen in Ascomycetes but also in Deuteromycetes. Remember, they are exogenous spores, they are asexual spores. That completes Ascomycetes, and I want to tell you a few examples. Eustylago is one example for that. Alright, which cause Aspergillus is an example which produce citric acid, and you have got claviceps ergot disease in rye, penicillium, which is responsible for the production of an antibiotic, Alexander Fleming, chain, florey, staphylococcus. That story is known to you. Morels and truffles are costly mushrooms. Alright, Neurospora, very popular example, an experimental member. These are all the examples of Ascomycetes. Even Monascus, which produce statins, which are cholesterol lowering agent, is also an yeast. Saccharomyces, used in baking and brewing industry, also belongs to Ascomycetes. Gibberella fusicorae, which cause Bacchianate disease in rice, is also an example of Ascomycetes member. Then we talk about Basidiomycetes. Now very simple, children. Basidiomycetes, these are the basidiospores. This is the primary mycelium. Now naturally, just like Ascomycetes. The only thing is that here in place of ascospores I call basidiospores. Again primary mycelium. There we see sex organs, and ascagonium and antheridium. Nothing like that. Directly the hyphae fuse. But again, just like in Ascomycetes, plasmogamy occur but not karyogamy, resulting in the formation of a dikaryotic mycelium. What next happens? This dikaryotic mycelium is going to produce aerial fruiting bodies, and these aerial fruiting bodies are called mushrooms or basidiocarps. And just like an umbrella, it has got a stalk and a cap on the lower side, like an umbrella. You see the presence of gills. So if you take a section of a gill, you see a club-like structure. That is the reason why called club fungi. A club-like structure called basidium. Now the 2 nuclei fuse. Plasmogamy is occurring here, karyogamy is occurring here. Just like in Ascomycetes, long time gap between plasmogamy and karyogamy. That is called sac fungi, club fungi. The 2 nuclei fuse, reduction division. The difference is these spores are produced externally, not internally. These are the 4 basidiospores which are produced externally. Then you may ask me, what are the asexual spores here? Are they conidiospores? No. Basidiomycetes does not show any asexual reproduction. Vegetative reproduction is seen. Eustylago cause smut, Puccinia cause rust, and you have got agaricus, which is the white button mushroom. They are the most common examples. Puffballs, all right, is also an example. Lycopteridon, bracket fungi, polyporus, they are all examples of Basidiomycetes. And I don't want to talk much about Deuteromycetes, children. Very easy one. It's called fungi imperfecti, only asexual stage, conidiophores, no sexual stage. If at all any Deuteromycetes member shows sexual stage, it is shifted to Ascomycetes with a separate name. And they reproduce asexually by conidiospores. They are major decomposers. Help in mineral cycling is a good question. Example, trichoderma. Trichoderma, where did we study? Microbes chapter we studied. Trichoderma is a biocontrol agent, eliminating root-borne fungal pathogens around the root system of higher plants. Trichoderma can also produce immunosuppressive agent called cyclosporin A. After organ transplantation, the immune system should be calmed down because a new device is introduced into the body. The immune system becomes very agitated. So you have to calm down that. For that we use cyclosporin A obtained from Trichoderma. Colitotrichum, red rot of sugarcane. Alternaria, early blight, they're all the ones which are caused by Deuteromycetes members. We don't have kingdom Plantae and Animalia, putting aside viruses directly. Ivanovsky called it as filterable agents because they're able to pass through bacterial filters. Biogenic, after B comes C, Contagium vivum fluidum, or coined the term virus, it's also starting with C. Stanley, for the first time, crystallized, been separated virus and studied them in detail, for which he got a Nobel Prize. Viruses shows both living and non-living characters. Living characters, they can multiply inside the host, they contain genetic material, either DNA or RNA, not both. Non-living characters, they're acellular, they doesn't have any metabolism, cannot respond to antibiotics. Then we have got two examples of virus, TMV and bacteriophage. TMV is a rod-shaped virus. Bacteriophage is a tadpole-shaped virus. In TMV, the genetic material is RNA, single-stranded RNA, whereas in bacteriophage, generally the genetic material is a double-stranded DNA. In TMV, all the capsomeres are arranged helically, helical symmetry. Whereas in bacteriophage, in the head, capsomeres are arranged in the form of 20 equilateral triangles, cubic symmetry. Whereas in the tail, the capsomeres are arranged in the form of a helix, helical symmetry. Two types of symmetries are seen in a bacteriophage. Head shows a cubic symmetry and the tail shows helical symmetry. And there are 6 tail fibers, also called prongs, which secrete an enzyme lysozyme, which can digest the bacterial cell wall, and the viral DNA can enter into bacterial cell. Capsid is a protein coat, don't forget, it protects the genetic material. So that's about viruses, children. We have got prions and virioids. Prions, they have only protein coat, they are not viruses. They are similar to viruses, they are acellular, they contain only protein coat. Protein coat is infectious, protein coat causes 2 types of diseases. If it is in cattle, mad cow disease or BSE, bovine spongiform encephalopathy. And if it is in humans, it is Creutzfeldt-Jakob disease. They are all neurological diseases which is caused by the infectious protein. Highly folded protein, prion, P, prion, protein, prion, easy way to remember. Virioid, they are normally infects plants unlike prions, citrus hexocortosis disease, potato spindle tuber disease, they are all caused by virioids. Dinah is a scientist, they have low molecular weight RNA that is capable of causing diseases. The only cellular organisms which did not find any place in Whittaker's classifications are lichens. You may ask me why? Because they show algal and fungal partners. The algal partners include green alga and what is called blue-green alga, and the fungal partners include Ascomycetes and Basidiomycetes. They are good pollution indicators. They are also the pioneer community for an ecological succession operating on rocks. Phytoplanktons are the pioneer community for an ecological succession operating in water, hydrosphere, and lichens and mosses are the pioneer community, first community for an ecological succession operating on rocks. That completes biological classification, which took 5 minutes more, children. I thought of 20 minutes, but I covered every point what is there in this. I'm pretty sure that you are properly placed here. There is no doubt. Now the last 10 minutes, all right, of the class where we have to get rid of plant kingdom. I know it's not so easy, but we'll try to complete it in last 10 minutes. Plant kingdom. Before we get into the plants, previously what are the organisms which are kept in plants? Now they are not there. What are list? Let us list out. Bacteria. Previously they are kept under plant kingdom. Now they are kept under Monera. Okay. Then next one, cyanobacteria. Previously they are kept under plants. Now they are kept under Monera. Okay. Next, chlorella and chlamydomonas. All right. Previously they are kept under plants. Now they are kept under Protista. Right. All right. Fungi. Previously they are kept under plants. Now they are given a separate kingdom called fungi. So your first paragraph talks about the organisms which are previously kept under plants are distributed into different families in the modern classifications. Then we have got artificial, natural and phylogenetic. Without wasting any time, artificial. It is based on both vegetative reproductive characters. It is based on few superficial characters. Organisms which are dissimilar are grouped together. Organisms which are similar are separated. That is the demerit of this classification. Examples: Linnaeus' sexual system of classification, which is based on the nature of stamens and rhizome. Alright, even Aristotle's classification of plants into herbs, shrubs and trees is artificial because it is based only on one gross superficial character, that is nature of stem. Alright, chalo. Next we go to natural. The meaning of natural classification, which is based on all the morphological characters, and not phylogeny, that is said to be natural. Both external, internal characters, anatomical, cytological, all are taken into criteria. Bentham and Hooker classification is a wonderful example for natural system of classification. Don't forget that. Natural affinities are taken to criteria. Organisms which similar characters are more or less grouped into the same unit. That is the biggest advantage. Demerit, they did not take evolution as a criteria. Bentham and Hooker, don't forget that classification. Next is phylogenetic. Organisms of the same taxon have a common ancestry. That is the belief of this phylogenetic classification. They not only took external, internal characters, but also evolution as the criteria, the tool for classification. It is the most acceptable classifications, phylogenetic. Whitaker's classification is phylogenetic. Not only that, Engler and Prantl, Hutchinson, alright, Dactogen, they're all classifications which are said to be phylogenetic. Chemotaxonomy, based on chemical constituents, you classify. Cytotaxonomy, based on chromosomal structure, behavior, you classify. Then numerical taxonomy, all the observable characters are given codes, and based on the number of overlapping codes, you keep all the plants in that particular group. So alga, the next unit children, alga. How much time we are going to take? 5 minutes, listen carefully. Habitat, they are mostly aquatic. Coming to the cell wall, they contain cell wall material, pectin and cellulose if it is a green alga, algin and cellulose if it is a brown alga, carrageen, polysulfate esters, agar-agar if it is a red alga. Phycocolloids are seen in the cell walls of brown alga and red alga is a very, very important point. Alright, then we talk about the plant body, thallus organization. It is more or less green in green alga, and if it is with a brownish tinge, if it is a brown alga because of fucoxanthin, and it's got red color phycoerythrin, so therefore red alga have got a reddish tinge. That's about thallus. Complex body organization is seen both in brown and red. Holdfast type and frond is seen in brown, and even in red you see what is called in polysiphonia, the organization is more complex. Coming to the chlorophyll pigments, A and B in green, A and C in brown, A and D in red. Coming to other pigments, carotenes are seen in green, fucoxanthin brown color in brown, and phycoerythrin in red. Okay, coming to flagella, 2 to 8 in green, good question, 2 to 8 in green, and in brown 2 lateral flagella in spores and gametes. Pyriform, 2 lateral flagella. In red, no flagella. Red means stop, not only in signal but also in alga, stop. Non-motile, so no gametes and spores are non-motile there. That's about the flagella. The next important one is sexual reproduction. Isogamy, anisogamy and oogamy in brown and green, and majority of members shows oogamy in what is called red. Then coming to asexual reproduction. Asexual reproduction is mostly by zoospores, chlamydomonas. In green alga, they reproduce asexually by zoospores. Almost all brown alga reproduce asexually by zoospores, pyriform, 2 lateral flagella, unequal. And in red alga, tetraspores, they are non-motile asexual spores. Non-motile asexual spores in red alga are called tetraspores. So asexual spores may be zoospores or tetraspores. Most of the cases, it is zoospores. Vegetative reproduction, fragmentation, it is distributed almost to all the 3 classes. What are the criteria for classifying alga into Chlorophaceae, Phaeophaceae and Rhodophaceae? It is based on the pigments and also the reserve food. The reserve food in green alga is starch, it is stored in pyrenoids. The reserve food in brown alga is laminarian starch and mannitol, and the reserve food in red alga is floridian starch, which resemble amylose and amylopectin. That point is important. And the economic importance, children, of alga, if you look, more than 70 genera are of edible nature, especially marine forms, sargassum, porphyra, laminaria, etc. Chlorella is used as SCP. Don't forget agar-agar, used in making the medium semi-solid, is obtained from Gelidium and Gracilaria. Don't forget algin is a cell wall material, water holding chemical present in the cell wall of brown alga. Don't forget carrageen is a hydrocolloid present in the cell walls of red alga. Cell walls of red alga also contain galactans, mannans, etc., which is there in the cytology unit. So that is the broad classification of alga, children. Now if you just spend 1 minute for each alga, green alga, 1 minute. They are mostly aquatic, mostly freshwater and marine. Thallus, unicellular green alga, chlorella and chlamydomonas. Volvox is colonial. Multicellular unbranched filament with isogamy and non-motile spirogyra. Multicellular unbranched filament, isogamy and motile eulothrix. And isogamy is seen in Eudorina. Oogamy is seen in Cara and Volvox. Finished. And very important points about green alga. Majority of green alga show a diversity with regard to the shape of the chloroplasts. Majority of green alga are what is called going to show isogamy, anisogamy and oogamy. In majority of green alga, the zygote undergo reduction division, therefore the haploid phase is dominant, haplonetic life cycle is very commonly seen. 2 to 8 flagella is equally important with regard to green alga chlorophacy. Outer pectin, inner cellulose. Finished. Then we go to brown alga. Except ectocarpus, which is simple and filamentous, the remaining all ones contain holdfast type and frond. Laminaria, the frond is edible. Alright, it is dichotomously branched with air bladders which help in buoyancy in almost all kelps. Kelps are nothing but all the massive plant body containing brown alga are called kelps. Cell wall, outer algin, inner cellulose, chlorophyll pigments A and C, fucoxanthin imparts brown color, reserve food, laminarian starch and mannitol, and asexual reproduction, zoospores, pyriform, and sexual reproduction, isogamy, anisogamy and oogamy. Very important. Sexual reproduction may occur inside a female sex organ called oogonium, or it may occur outside in water. External fertilization is very unique of alga. Alright. Then we talk about red alga. Porphyra is red alga which is edible. Polysiphonia is highly branched. Red alga contain agar-agar, polysulfate esters, carrageen in their cell walls which are hydrocolloids. They contain chlorophyll A and D. Red color is because of phycoerythrin. They contain floridian starch which resemble amylose and amylopectin. Asexual reproduction is by non-motile spores called tetraspores. Sexual reproduction is mostly oogamous. Post-fertilization changes are complex. They can stay either in peripheral waters or deep waters which are warmer. That's about red alga. Porphyra, Polysiphonia, Gelidium, Gracilaria are the most common examples of red alga. They mostly show oogamy. Post-fertilization changes are complex. Agar-agar obtained from Gelidium. Carrageen is all these are very important points about. Alga is the most fertile part for asking questions in NEET for the last 2 or 3 exam, children. That is the reason I took a little more time to discuss about alga. Coming to bryophytes. Before I go to bryophytes, what are distinctly seen in alga? Only in alga. Haplonetic life cycle, only in alga. External fertilization, only in alga. Chlorophyll A, B, C, D, only in alga. Different shapes of chloroplast, only in alga, that too in green alga. Different compositions of the reserve food, laminarian starch, mannitol, floridian starch, that is also very unique of alga. Alright, the plant body is filamentous. These are all the very important points with regard to alga. Please don't forget that. Now we go to bryophytes. Now from here onwards, we are not going to take much time, 2 minutes for each group. Now, the first bryophyte, what we're going to talk about is liverworts. I told you in my previous class, please go and watch one more video on Plantkingdom which I made. Alright, where I explain things very slowly, how to remember, I said. Go with the table, I said. What is the table? First is gametophyte. This is a gametophyte. This is a gametophyte which contain unicellular rhizoids, which also contain gemmae, which are multicellular green asexual buds. This is Merkensia. This is the gametophyte. What is the meaning of gametophyte? Phyte and a plant producing gametes. So it should produce gametes. It contains a long stalk, and on this you see what is called a male sex organ called antheridium, and this male sex organ produce what is called male gametes. Finished. That's very easy. Over. This is one more gametophyte. It should produce gametes. It contains a female sex organ called archegonium. Already I told you. In alga, the female sex organ is oogonium in brown alga, not archegonium. Archegonium is seen for the first time in bryophytes. So gametophyte is over. Gametangium is over. Gium means bag. This is the bag which produces gametes. Male gametangium, antheridium. Female gametangium, archegonium, which contain an egg cell. So gametophyte, gametangia, gametes. Now what should happen? Gametes fuse to form zygote, yes. Zygote develops into embryo, yes. What should embryo develop? This embryo will develop into sporophyte. This is the sporophyte. Foot, seta and capsule. Look, where is this sporophyte? This sporophyte is attached to gametophyte, which is very unique of bryophytes. It is nutritional and physical attached to gametophyte is very unique. What is you see in bryophytes. Now what does the sporophyte will do? It's common sense. Sporophyte should produce spores. This sporophyte will have a sporophyll. This sporophyll will have a bag called sporangium. This sporangium contains spore mother cells. They undergo reduction division and produce spores. 50% of spores will develop into a gametophyte which antheridia, and the remaining 50% develop into a gametophyte with archegonia. I repeat again, gametophyte, male gametangium, male gametes, female gametangium, female gamete, zygote, embryo, sporophyte, sporophyll, sporangium, spore mother cell, spore, and spore again produce gametophyte. This is Mercantia, children, liverwort. The reason why it's called liverwort is because the gametophyte looks like the lobes of the liver, hence you call them as liverwort. Then we talk about a moss, all right. This is a gametophyte. Initially, the gametophyte is juvenile, filamentous. You call them as protonema. Why I'm starting with gametophyte? Because in alga, bryophytes, it is the gametophyte which is very well developed and dominant, free living. All right, this is a free living green gametophyte. Even in Mercantia, the gametophyte is green and free living. It is the sporophyte which is a parasite, not gametophyte. This is the gametophyte called protonema. There are some protonema which bear buds, you call them a secondary protonema. Those secondary protonema germinate and give rise to a leafy and adult gametophyte. This leafy and adult gametophyte is called gametophore. At the tip of it, you see what is called antheridia. And at the tip of the lateral branch, you see what is called archegonia. Finished. From here on, the remaining story is same. The only difference between Mercantia and Funaria is, in Mercantia, the gametophytes are unisexual, whereas here, the gametophyte is bisexual. This is young gametophyte, adult gametophyte, which is bisexual, producing antheridia and archegonia. The remaining story is known to you. In the presence of water, they fuse to form a diploid zygote. Zygote develops into embryo. Embryo develops into sporophyte, foot, seta and capsule, sporophyll, sporangium, spore mother cell. Spores get liberated. The remaining story is known to you. Polytrichum is a hairy cap moss. Sphagnum has got some economic importance. Normally, bryophytes don't have a huge economic importance. Sphagnum, it can be used as fuel. It can be used as packaging material. All right, that's about bryophytes, children, which is looking very fine. Mercantia, liverwort, and Funaria, moss. In Mercantia, you have seen gametophytes unisexual. Here it is bisexual. In Mercantia, no protonema. Here you see protonemal stage. In Mercantia, gemmae are there. There are no gemmae. Secondary protonema there, which help in vegetative reproduction. All right, in Mercantia, the gametophyte looks like the lobes of the liver, whereas in Funaria, young gametophyte is thalloid and adult gametophyte is leafy. In Mercantia, antheridia and archegonia are present on separate gametophyte, whereas here, antheridia and archegonia is the same, therefore it is bisexual. The remaining story is same, children. This is looking fine. That completes this next unit, that is said to be bryophytes. Now we have the last two groups, pteridophytes and gymnosperms. Pteridophytes 2 minutes, gymnosperms 2 minutes, not more than that. Pteron is equal to feather, phyta is equal to plant. Plants with feather like leaves, you call them as pteridophytes. First time in pteridophytes you see vasculature, therefore they are rightly called vascular cryptogams. Alright, now if we talk about pteridophytes, you have got Salaiopsida which are extinct, Salayotum, Lycopsida, Salaginella, we have got Spinopsida, Equisetum, and you have got Pteropsida ferns. Shall I tell you, in your book there are 4 diagrams? Alright, in your book there are 4 diagrams here, here, here and here. Yes or no? This is Salaginella, this is Equisetum, this is ferns, and this is Salvinia. Now listen, I'll tell you some easy way to remember. Salaginella and Salvinia, these two, they are heterosporous and also heterophyllous. 2 types of leaves, heterophyly, cotton, larkspur, coriandrum. You studied that under plasticity of plant growth. Apart from that, these two are also heterophily. Salaginella and Salvinia. Heterospory, many precursor characters to seed habit, and also heterophily. Very easy. Then, what about these two? Salaginella and Equisetum, they contain strobilus. They contain strobilus. All right, that is Salaginella and Equisetum. The next important one we have is what is called Equisetum and ferns. Equisetum and ferns, they are homosporous. These two are heterosporous, and these two are examples for homosporous. Easy way to remember. And last important point, Salaginella show microphylls, and what is called fern shows macrophylls. The leaves are very big. And the last minute, children, we are going to spend here coming to what is called ferns. This is the sporophyte, yes. This is a sporophyll, yes. Sporophyll will have sporangia, yes. Inside that spore mother cells, yes. Reduction division spores. Spores get liberated. The spores develop into gametophyte, which is called prothallus. Sporophyte can grow on all types of soils because it has got vasculature, not the gametophyte. It doesn't have vasculature. But the important thing is that both of them are independent, attached to soil and green, which is unique feature of pteridophytes. Just because it's gametophyte cannot grow on all types of soils. Sporophyte is restricted to narrow geographical regions. One more reason is this gametophyte produce anthereria and archegonia. Fertilization also require water, not only in alga, not only in bryophytes, but also in pteridophytes. That is the reason the restriction, the growth of sporophyte is restricted to narrow geographical regions. Again, I repeat, sporophyte, sporophyll, sporangium, spore mother cell, spore, gametophyte, male gametangium, female gametangium, multiciliated male gametes fuse to form zygote. Zygote develops into embryo, and embryo is going to develop into a sporophyte. Salaginella show many precursor characters to seed habit. Remember that, along with Salvinia. Coming to the gymnosperms, last 10 statements for gymnosperms, only 10. Root system, habitat, majority of them are xerophytes. 2. Habit, majority of them are trees. Root system, coralloid roots are there in Cycas, mycorrhizal roots are there in Pinus. Stem system, stem is unbranched in Cycas, stem is branched in Pinus, Cedrus and Jingo. Leaves, leaf shows many xerophytic adaptations, xerophytic characters. In Pinus, the leaves are reduced to needle-like structures, thick cuticle, sunken stomata, very popular question. Many xerophytic adaptations you see in leaf. And coming to what is called sexual reproduction, children, remember I'll tell you 5 points. There are cones in place of flowers. Remember, if it is loosely attached, you call them as lacs. Alright, so they are not called flowering plants because in place of flower, cones are there. Cones contain microsporophylls, means comparable to stamens. They contain microsporangia. Microsporangia produce microspore mother cell, produce microspores. Microspores are called pollen grains. They get dispersed by wind, anemophily. These pollen grains directly reach the ovule because there is no ovary style and stigma. They directly reach the ovule. Pollinating agent is wind. And there what happens? The pollen tube is produced from vegetative cell. Then the generative cell divide and produce the 2 sperms. Just like in angiosperms. The sperms are carried up to the mouth of archegonium because of the pollen tube, which is called siphonogamy. And last 5 points. The female reproductive system. Female plant is there in Cycas, unlike Pinus, where male and female cones are there on the same plant. Cycas, male and female cones are separate plant, dioecious. A female plant, megasporophyll is open. There is no ovary style and stigma. Ovules are naked. Megasporangium, one of the nucellate cell, megaspore mother cell. Reduction division, 4 megaspores, 3 megaspore disintegrate, functional megaspore. It develops into a female gametophyte called endosperm. Interesting, endosperm is formed before fertilization. It's a pre-fertilized product. It is haploid, very important. Few cells on the top transform into archegonia. Archegonia. So archegonia inside the ovule is very typical of gymnosperms. Once the pollen tube extends, diploid zygote, embryo, after embryo, this naked ovules, which now become seed, are dispersed, and 50% of them will develop into male and 50% of them develop into female. Remember, in gymnosperms and angiosperms, siphonogamy is seen. Remember, in gymnosperms and angiosperms, seeds are seen. Remember, in gymnosperms and angiosperms, you see gametophytes highly reduced, unlike sporophytes, which are very well developed. These are the important points with regard to plant kingdom. And life cycle in gymnosperms and angiosperms is diplonetic. In alga, haplonitic. In bryophytes and pteridophytes, haplodiplonetic. Whereas in gymnosperms and angiosperms, the diploid phase is dominant. Therefore, the life cycle is diplonetic. That completes this topic. Remember these points thoroughly, and that will help you to recollect all the points on the day of exam. We'll meet tomorrow with plant physiology and cytology. Thank you.